I apologize for this short little video. My microphone decided to go out in the middle of my last recording, and I decided not to start the whole thing over. So this one will be a little bit short. We're going to wrap up humans and disease and the use of biotechnology. So let's start over on this slide. So if gene therapy is so fabulous and biotechnology is so great, why have we had a poor record of success in curing diseases? Well, there are several reasons for this. The first being that it's really hard to get a working gene into specific cells. You have to get them in the right cell and only that cell. There's another problem with getting enough of the gene into enough, or a copy of each gene, into enough cells that you're actually going to have a physiological effect great enough to cure the disease. And then you also have the unintended consequences of transferring a gene into unintended cells. Now, CRISPR might actually be able to take care of all of those things because CRISPR is so fantastically precise, and the methods for actually getting CRISPR into cells are becoming more widely available and more efficient. But there's also the difficulty in regulating gene expression. So once you get the gene into the cells that you need, you also have to have that gene expressed. And this is another problem. I'm not actually going to go into this because this is a much more detailed molecular explanation than we need to go to here. But this is another problem that is facing, that gene therapists are facing with their current biotechnology practices. So we've talked a little bit about cloning. Cloning can mean making copies of genes, it can mean producing organs, and it can also mean producing individuals. So probably most of you think of cloning in reference to a whole organism. So let's talk about what that actually means. Now we've already talked about cloning genes, and you can use that for a variety of purposes, but at the whole organism level, this means that you have created a new individual that has exactly the same genome as another individual. Now this is not the same thing as producing an identical twin, and the reason is that these individuals are not from the same parent, so to speak. They are separated in time, so maybe by months, days, even decades. So you do this by isolating an egg from whatever organism you want to clone. Let's look at sheep. The most famous example, the first example, was a sheep called Dolly in the United Kingdom. And you take the egg from one sheep and a mammary cell from another. So the egg is going to be the recipient of the genetic information. The mammary cell is going to be the source of the genetic information. So you're going to remove the nucleus that contains the DNA from each of those cells, and you're going to insert the nucleus from the mammary cell, from the donor organism, and put it into the egg of the other organism. So then you are going to induce that egg to divide through normal early development until it becomes an embryo. Then you're going to transplant it into the uterus of a surrogate sheep, and that sheep is going to give birth to the cloned sheep. So that cloned sheep is genetically identical to this sheep that donated the mammary cells. So there's a couple other examples. We've got some cats and some rabbits. This has been done with a few different organisms throughout the years. The Dolly was produced in 1997, and the researcher Ian Wilmot was a British scientist who got his idea from an earlier scientist who proposed that this was possible. Initially it was difficult and inefficient, and out of 272 tries they only got one successful surviving organism. But it's become a bit better. And as I said, several different organisms have been produced through cloning. So at some point, cloning humans is possible, but in general, I think most people think that this is a bad idea. It is certainly not supported by the scientific community. And of course, we could go into ethics all day about whether or not you should do that in animals or in humans. So biotechnology has been really important for the field of forensics because DNA can be used as an individual identifier. So this allows people to be identified based on their unique gene sequence. So all individuals have their own unique gene sequence, and forensic science makes use of something called an SLP. Now, an STR, I got my acronyms mixed up. And this is a short tandem repeat. And we'll talk about those on the next slide, but basically all individuals differ in their number of repeats. And that allows you to take what we call a DNA fingerprint. And you can use restriction enzymes to produce a very specific pattern in your DNA. So this guy here, Colin Pitchfork, was an individual who was living in England in the 1980s. And initially, he was probably going to walk free from a murder and rape conviction. But a British biologist named Alex Jeffreys made a discovery that small pieces of DNA within everybody's chromosomes vary tremendously. And those fragments vary enough that they're unlikely to be identical between two people. And so this man was charged with murdering and raping 2 high school girls in his village. A different man confessed to one of the murders, but the police department didn't believe that he actually did it because the crimes were so similar. And so they actually went through the area and requested blood samples of all the men between 18 and 35, looked at more than 5,000 blood samples, and were ultimately able to convict him of this crime. Now, according to our book, he actually initially asked a friend to give him a blood sample for him, and police found out about it. And so he was the first person to be convicted of a crime based on DNA evidence. So let's talk about how this works. So among any 2 individuals, 99.9% of the sequence is identical. But that 0.1% of the roughly 3 billion base pairs that we have in our genomes works out to approximately 3 million base pair differences between any 2 people. Now, the exceptions are identical twins, but if we exclude them, all people are going to be genetically different in specific regions of their DNA. So this region are regions called short tandem repeats, or STRs, and these are regions where a small sequence repeats over and over and over again. And these STRs vary greatly among individuals. And so you can use this as sort of a marker to see the difference between 2 people. So if we look at this diagram here, individual A has a particular sequence of short tandem repeats. Mother and father are actually different in this location for that individual's mother and father. And so this individual is going to have a very specific pattern of repeats. This individual down here has a different number of repeats coming from each parent, and so they are going to have their own pattern of repeats. You can use this DNA fingerprint to determine whose DNA or whose sample belongs to who. You can use it for paternity testing. And you do this by creating essentially a DNA fingerprint. So you use PCR to amplify a region that is high in short tandem repeats. And when you do that, you get a sample that has different lengths. So this individual here who had a short set from her mother and a longer set from her father is going to produce two bands of DNA, one amplified for the larger fragment and one amplified for the smaller fragment. Now then you can visualize this using something called gel electrophoresis, which we will talk about in lab, but you can compare that to somebody else's, because this individual here will have a different sized smaller fragment and a different size larger fragment, and so you can get individually individual fragments for each person, and those fragments are going to be unique to each person. Now in lab we are going to use this gel electrophoresis, and gel electrophoresis is a technique in which you can separate DNA based on size. So you will have what we call a gel. It's essentially like a real thick jello, and you apply a current to it. So you put your DNA sample into the gel, into a little well in the gel, and DNA itself is negatively charged. So when you apply a current, that DNA sample is going to run towards the positive end of your chamber and pull the sample, spread it out. Now it's going to move based on charge, but it is also going to move more slowly depending on how large it is. So small pieces are going to move farther, larger pieces are going to move more slowly, and so you end up with a pattern of DNA.